Hey everyone, welcome back. Ready for another deep dive? Today we're going deep into SMAD-independent pathways.
Oh, yeah, these are exciting.
They are. We've got some really interesting research papers and even a YouTube video all about how these alternative routes, especially those with MAPK and Rho,
Mm.
really change up how cells respond to the whole TGF-beta superfamily.
Yeah, it's like we're going to discover a whole secret communication system happening inside our
Right.
cells.
It's so cool. And we'll be talking about all the big questions like who are the important players in these pathways and what even turns them on?
And how do they sometimes work with and sometimes totally fight against the normal SMAD signaling?
Yeah, and why is it even important to understand these non-SMAD pathways anyway? Especially when we think about all the different things TGF-beta does.
It does a lot.
Okay, so let's get started with something that really puzzled scientists for a while.
What's that?
It's about those heart muscle cells. You know, the ones that keep our blood pumping those cardiomyocytes.
Ah, yes, the cardiomyocyte mystery. This one really shook things up.
It did. So this YouTube video, Cyclic AMP Signaling Part 9, talks about how if you stimulate two different receptors on these heart muscle cells,
Okay.
the beta-1 adrenergic receptors with adrenaline
Mm.
and the EP4 receptors with prostaglandin E2, they both end up making cyclic AMP.
And cyclic AMP is like this messenger molecule inside cells, right?
Yeah, it's really important. But here's the thing. Scientists used to think that cyclic AMP just kind of flooded the whole cell and affected everything equally.
Hmm.
So if that was true, then why does only adrenaline and not prostaglandin E2 make the heart muscle contract more forcefully? They both trigger cyclic AMP, so it gives.
Yeah, that's where the old way of thinking just fell apart. It couldn't explain why one was specific and the other wasn't.
Right.
And that's actually what led to the idea of localized signaling. Like the signals are actually compartmentalized within the cell. They don't just go everywhere.
Oh, so it's like instead of sending a flyer to everyone in the city, you're sending a targeted letter.
Exactly.
Right.
Much more efficient.
That makes so much more sense. But then how do cells manage this precise signaling? It seems complicated.
Well, that's where these really cool proteins come in. They're called akinase anchoring proteins, or AKPs for short.
The video calls them scaffold proteins.
Mhm.
What does that even mean?
Okay, imagine you're on a construction site.
Okay.
You need scaffolds to hold all the materials in place right. Well, AKPs are like tiny scaffolds, but inside cells.
Well,
They hold a key enzyme, protein kinase A, or PKA, in very specific spots.
and the video mentioned there are over 50 different AKPs in humans. That's a lot of scaffolding.
It really is, and that just shows how important this whole precise localization thing is. You need all these different scaffolds to hold things in the right places for the cell to work properly.
So it's not just about sending the message, but also about making sure it gets to the right department in the cell.
Exactly, you got it. Each AK has a targeting domain that's like a postal code sending it to the right spot in the cell, and then there's the PKA binding domain, which does exactly what it sounds like. It holds on to PKA and keeps it inactive until it's needed.
So it's like PKA is on standby, waiting for its cue to jump into action at a very specific location.
Yeah, exactly. That's a great way to think about it.
Wow, cells are amazing. The video also mentions something about the structure of AKPs and this thing called an amphipathic helix.
Oh, yeah, those are cool. They might sound complicated, but they're actually a really elegant solution.
Okay, break it down for me. What is an amphipathic helix?
It's basically a helix with two sides, one that loves water and one that hates it.
A helix with a split personality.
Kind of, but it's this unique structure that lets the AKP bind to PKA and anchor it to that scaffold.
So it's like this perfect molecular handshake, making sure everything stays in place.
Exactly. And it's this precise anchoring that makes that localized signaling we were talking about earlier actually work.
This is making me think about how much we still don't know about cells. Well, sir. There's always something new to discover.
Absolutely. There's a whole universe inside each one of our cells, and we're only just beginning to explore it.
I know, right? Okay, let's switch gears for a bit and talk about another example that shows how these alternative pathways work. Thyroid hormone receptors, or TRs. They're like tiny switches inside cells that control whether genes are turned on or off.
Oh, yeah, TRs. They're fascinating. What's really cool about them is that they can actually act as both a gene silencer and a gene activator.
Wait, so they can turn genes both on and off?
Exactly. It all depends on whether a thyroid hormone called T3 is bound to them.
This thyroid hormone receptors paper describes them as molecular switches, which I think is a perfect analogy. So when T3 isn't around, they actually silence genes.
But then when T3 binds, boom, it flips the switch and those genes get turned on.
It's like magic. But the paper also mentioned that the type of DNA sequence these receptors bind to can also play a role in whether they activate or repress gene expression.
Right, so it's not just about T3, it's also about the context of where the receptor is binding on the DNA.
Wow, that adds another layer of complexity.
It really does. It's like the receptor is reading the instructions on the DNA to figure out what to do.
Cells are so smart.
They really are. And if that wasn't complicated enough, these TRs also have a whole team of supporting proteins called core pressors and coactivators.
It's like they have a whole entourage. What do these core pressors and coactivators actually do?
Well, imagine them as the stage crew and lighting technicians for a play.
Okay.
They're crucial for setting the stage for gene expression. They either help silence genes or activate them depending on the cues they receive.
So what happens when T3 isn't around?
In that case, the core pressors bind to the TRs and bring in enzymes that modify histones.
Remind me again, what are histones?
They're the proteins that DNA wraps around,
Yeah.
like thread on a spool.
Right, right.
So these modifications basically make it harder for the machinery that reads genes to access the DNA. It's like putting a lock on the door to the gene.
So the gene is basically shut down.
Exactly. But then when T3 binds to the TR, those core pressors detach and the coactivators step in with their own set of enzymes.
What do they do?
They do the opposite. They make the DNA more accessible. So like they're unlocking the door to the gene and letting the show begin.
So the core pressors are like the bouncers and the coactivators are like the ushers welcoming everyone in.
Perfect analogy. And this paper also highlights a specific part of the TR protein called helix 12. It's kind of like a lid on the hormone binding pocket.
Go.
When T3 binds, the position of helix 12 changes, and that little shift is what kicks out the core pressors and lets the coactivators bind.
So a tiny structural change has a huge effect on what the protein does.
Exactly. It's amazing how these tiny changes can have such big consequences inside a cell.
It really is.
Yeah.
The paper also talked about some real-world examples of how these things can go wrong, like with a disease called resistance to thyroid hormone, or RTH.
RTH is a great example of what happens when mutations in the TR gene mess up this delicate dance of signaling.
So what happens?
Well, sometimes these mutations directly affect helix 12, preventing it from working properly.
So even when T3 is present, the core pressors can't be released and the genes can't be activated.
Exactly. It's like the switch is stuck in the off position.
That sounds like it could cause some serious problems.
It definitely can. The paper also mentioned something called the Vir-A oncogene, which is a mutant form of TR that doesn't even have helix 12.
And oncogenes are the ones that can cause cancer.
That's right. And because this one lacks helix 12, it can't release core pressors, so it's constantly silencing genes even when it shouldn't be.
Oh, wow. That's scary. So it's like this rogue gene silencer running around causing trouble.
Pretty much. And this can really disrupt normal cell growth and potentially lead to cancer.
It's crazy to think how something so small can have such a big impact on our health.
It really highlights how important these tiny molecular mechanisms are for keeping our bodies functioning properly.
Okay, before we move on, the paper teased us with the idea that TRs might have functions beyond just hanging out in the nucleus.
Ah, yes, the non-genomic actions.
They're like secret agents working behind the scenes. What do we know about these non-genomic actions?
It's a pretty new area of research, but it seems like TRs might actually be interacting with other proteins outside of the nucleus, maybe in the cytoplasm?
So they're not just controlling gene expression.
Nope. It seems like they might also be influencing things like cell signaling and how things are transported within the cell.
Wow. So they have a side hustle. It's like they're moonlighting in other parts of the cell.
That's a great way to put it. And it just shows us that even though we've learned a lot about TRs, there's still so much more to discover.
It's like peeling back the layers of an onion. There's always another layer underneath.
Exactly. And that's what makes this field so exciting. There are always new mysteries to unravel.
This is all so cool. I'm starting to see why understanding these alternative pathways is so important.
Me too. It really opens up a whole new way of thinking about how cells work.
Okay, so we've explored the mysteries of localized signaling with AKPs, and we've delved into the double life of TRs as molecular switches. Now I'm curious, how do these examples connect back to our main focus on SMAD-independent pathways and the TGF-beta superfamily?
That's a great question. And the key here is realizing that cell signaling isn't just a simple one-way street. There are multiple routes that signals can take, and these routes can even influence each other.
So instead of one clear path, there's this whole network of interconnected pathways.
Exactly. And just like with those thyroid hormone receptors, where the non-genomic actions add another layer of complexity, as MAD-independent pathways add a whole new dimension to TGF-beta signaling. They give the signals alternative routes to travel, which expands the possibilities for how the cell responds.
So they're like side roads or shortcuts that allow TGF-beta to get its message across in different ways.
That's a great analogy. These pathways, often involving kinases like MAPK and Rho, allow TGF-beta to regulate a wider range of things like cell growth differentiation and even cell death.
So it's not just about whether TGF-beta is present or absent. It's about which pathways are activated and how they all work together.
Exactly. And it's this complex interplay between all these different signaling pathways that makes TGF-beta such a powerful and versatile regulator of what happens inside cells, both in normal development and in disease.
Okay, I'm starting to see the bigger picture here.
Yeah.
These alternative pathways aren't just minor details. They're crucial for understanding the full scope of what TGF-beta can do.
Precisely. And that knowledge is essential for developing new therapies that can target these pathways and potentially treat diseases where TGF-beta signaling goes haywire.
That makes a lot of sense. But how do scientists even begin to untangle this crazy web of interactions? It seems like a huge challenge.
It definitely is. But researchers are using a variety of cool approaches to figure it out. One way is to use advanced imaging techniques that let us actually see these signaling events happening in real time.
So it's like watching a tiny molecular movie.
Exactly. We can see how these different proteins interact with each other and how their location in the cell changes in response to different signals. Another powerful tool is genetic manipulation, where researchers can turn specific genes on or off to see how it affects the whole signaling pathway.
So they can isolate the role of individual players and see how they contribute to the bigger picture.
Right. And by combining these and other techniques, scientists are gradually piecing together this intricate puzzle of TGF-beta signaling and all its different facets.
It's amazing how much progress they've made in understanding these complex pathways.
It is. And it's all thanks to their hard work and dedication to uncovering the secrets of cell signaling.
Well, I don't know about you, but my brain is feeling a little full right now.
Me too. We've covered a lot of ground today.
We have. But I think we've laid a solid foundation for understanding how these SMAD-independent pathways expand the influence of the TGF-beta superfamily.
I agree.
So before we dive even deeper into the specifics of MAPK, Rho, and all the other players involved, let's take a moment to let all this information sink in. We'll be back soon to explore those pathways in more detail and unravel their roles in both normal development and disease. Stay tuned.
Can't wait. Welcome back. Ready to jump back into those SMAD-independent pathways?
Absolutely. Let's untangle this web of signals. Before the break, we were talking about how these alternative pathways, often using kinases like MAPK and Rho, give TGF-beta more ways to control what happens inside cells.
Yeah, those options are really important for understanding how TGF-beta can have such different effects on different types of cells.
Okay, so let's talk about these other players. You mentioned MAPK and Rho. What do they actually do, and how are they different from the usual SMAD signaling?
Well, MAPK stands for mitogen-activated protein kinase. It's a whole family of proteins that are super important for transmitting signals related to cell growth proliferation and even how cells respond to stress.
So they're kind of like a rapid response team ready to jump into action when the cell gets a signal.
Exactly. And when it comes to TGF-beta signaling, MAPK pathways can be activated directly by the TGF-beta receptors or indirectly through other signaling molecules.
So there are multiple ways to get these MAPKs going. What about Rho? What does it do in this whole signaling network?
Rho or Rho-GTpases are another family of signaling proteins, but they're particularly important for regulating the cytoskeleton. You know, that internal scaffolding that gives the cell its shape and lets it move around.
Oh, right, the cytoskeleton.
Yeah. So Rho proteins are key for controlling things like cell migration adhesion and even cell division.
So both MAPK and Rho pathways sound pretty important, but I'm still a little confused about how they sometimes work with and sometimes against SMAD signaling. It's like they had this on-again, off-again relationship.
It is complicated, but we're starting to figure out some of the patterns. Sometimes these SMAD-independent pathways like MAPK and Rho work together with SMAD signaling. They can amplify the signal or make the cell's response more specific.
So they're like teammates working towards a common goal.
Exactly. But other times, these alternative pathways can actually block SMAD signaling, which leads to a totally different outcome.
So it's like a tug of war with each pathway pulling in a different direction.
That's a great way to think about it. And what determines whether they cooperate or compete? Well, that's something researchers are still trying to figure out. It probably depends on a bunch of factors like the type of cell, the specific TGF-beta ligand involved, and even what other signals are present.
So the context really matters. It's not a one-size-fits-all situation.
Exactly. And that's part of what makes this field so fascinating. There's always more to learn and discover about these interactions.
You mentioned earlier that understanding these non-SMAD pathways is especially important when we think about all the different roles TGF-beta plays. Can you expand on that a bit?
Sure. As we've been talking about, TGF-beta is involved in a ton of different cellular processes, everything from cell growth and differentiation to immune responses and wound healing.
And it's also been linked to various diseases like cancer, fibrosis, and autoimmune disorders.
Right, so it's a really powerful molecule with the potential to do both good and bad things. And because these SMAD-independent pathways add to the ways TGF-beta can signal, they also play a role in both its beneficial and harmful effects.
Okay, so these alternative pathways aren't just minor side routes. They're crucial for understanding the full range of what TGF-beta can do.
Exactly. And knowing this is super important for developing new treatments that can target specific pathways. Maybe we can enhance the good pathways and block the bad ones, depending on the disease.
So it's like having a more precise control panel for adjusting TGF-beta signaling instead of just turning it on or off.
That's a great way to think about it. And I think that's where the real potential lies in this field. By figuring out the complexities of these signaling networks, we can create new therapies that could really make a difference for people's health.
It's amazing to think about how much potential there is in this area of research. But how do scientists even begin to unravel all these complicated interactions? It seems like a massive undertaking.
It is a challenge, but researchers are using some pretty cool techniques. One approach is to use advanced imaging that allows us to actually see these signaling events as they happen in real time.
Wow. So it's like watching a tiny molecular movie inside the cell.
Exactly. We can observe how these different proteins interact with each other and how their location changes depending on the signals they receive. Another powerful tool is genetic manipulation, where scientists can switch certain genes on or off to see how it affects the whole pathway.
That's pretty cool. So they can basically isolate the role of each player and see how it contributes to the whole network.
Precisely. And by combining these and other techniques, researchers are slowly putting together the pieces of this TGF-beta signaling puzzle. It's a slow process, but they're making incredible progress.
This is all so fascinating. I can't believe how much is happening inside our cells at any given moment.
I know, right? It's like a whole universe inside each one of us.
Okay, so we've been focusing a lot on those SMAD-independent pathways. But let's shift gears for a bit and go back to those thyroid hormone receptors, or TRs. We talked about how they're like molecular switches controlling whether genes get turned on or off. But this paper goes into even more detail about their structure and how they actually work.
TRs belong to this really cool group of proteins called the nuclear hormone receptor superfamily. They're basically transcription factors, which means they control which genes are expressed in a cell.
And they usually have this modular structure, right? Kind of like building blocks that fit together to make a functional protein.
Exactly. Each domain or building block has a specific job. In the case of TRs, there's the DNA binding domain, or DBD, which allows the receptor to attach to specific DNA sequences near the genes it regulates.
So it's like a key that fits into a specific lock on the DNA, giving the receptor access to that gene.
Perfect analogy. And then there's the ligand binding domain, or LBD, which is where the thyroid hormone T3 binds.
Right. And we talked about how when T3 binds, it causes those shape-shifting changes in the receptor, like the movement of helix 12, and that ultimately determines whether the receptor silences or activates the gene.
Yeah, the LBD is like the sensor that detects whether T3 is present or not. And remember that the paper also mentioned that the specific DNA sequence the TR binds to can also influence whether it activates or represses gene expression.
Oh, right. So even with the same signal, the outcome can be different depending on where on the DNA the receptor is binding. It's like the receptor is reading the instructions on the DNA to decide what to do.
Exactly. And that just shows how much control there is within cells to make sure the right genes are expressed at the right time and in the right amount.
Okay, now let's dive a little deeper into those core pressors and coactivators. We talked about how they're like the supporting cast in the TR show, but what do we know about the specifics of how they work?
They're essential for fine-tuning gene regulation. Remember we discussed how core pressors like NCOR and SMRT help TRs silence genes by bringing in enzymes that modify histones and make the DNA harder to access?
Yeah, they're like closing the curtains on the show.
Right. But the paper also mentioned that core pressors can directly interact with the machinery that transcribes genes, so they can block gene expression from multiple angles.
Wow, so they're really making sure those genes stay silent.
They are. But then when T3 binds to the TR, those core pressors detach, and the coactivators come in with their own set of enzymes.
And what do the coactivators do?
They not only make the DNA more accessible, but they also interact with other parts of the machinery that's involved in transcribing genes. They help assemble the protein complexes that are needed to activate gene expression.
So they're like setting the stage and getting the band ready to play.
Exactly. And the paper also reminded us that there are two main types of coactivator complexes, ones that have histone acetyltransfer activity and ones that don't.
Right. Even more layers to this whole gene regulation dance.
It's amazing how complex it all is. We also talked about helix 12, that little lid on the hormone binding pocket, and how even a small change in its position can totally change what the receptor does.
It's mind-blowing how a tiny structural change can have such a huge impact inside a cell.
It really is. And remember those real-world examples we discussed, like resistance to thyroid hormone or RTH?
Where mutations in the TR gene, sometimes affecting helix 12, prevent the receptor from working properly.
Right. And we also talked about the VRBA oncogene, which is a mutant form of TR that doesn't have helix 12 at all, that leads to constant gene silencing and can even contribute to cancer.
It's incredible how something as small as a single helix can have such a huge effect on human health.
It really highlights the importance of these precise molecular mechanisms for keeping our bodies functioning the way they should.
Okay, before we move on from TRs, remember that little tease about them maybe having functions outside the nucleus? Those non-genomic actions?
Oh, yeah, those are intriguing.
It's like they have a secret life outside of controlling gene expression.
It does seem that way. It's still a pretty new area of research, but there's growing evidence that TRs might interact with other proteins in the cytoplasm, potentially affecting things like cell growth, metabolism, and even immune responses.
So they're not just stuck in the nucleus pulling the strings of gene expression. They're out in the cytoplasm getting involved in other cellular activities too.
Exactly. And some scientists believe that these non-genomic actions might even be faster than their regular gene-regulating activities, allowing cells to respond more quickly to changes in thyroid hormone levels.
So it's like they have a direct line to some cellular processes without having to go through the whole process of changing gene expression.
That's a great way to think about it, and it just goes to show that we still have a lot to learn about TRs and all the amazing things they can do.
It seems like every time we answer one question in biology, 10 more pop up.
That's the beauty of science. It's a never-ending journey of discovery.
I love that. Now, before we wrap up our exploration of SMAD-independent pathways and their connection to TGF-beta, I have a question for you. Do you think there are any similarities between these non-genomic actions of TRs and the actions of those SMAD-independent pathways we've been discussing?
That's a really interesting question. I think there could be some parallels. Both of them involve expanding the possibilities for signaling beyond the traditional pathways that go through the nucleus. They both allow cells to have more diverse and nuanced responses.
So they both add to the complexity of the signaling network.
Exactly. And I think there's a lot of potential for future research to explore how these different types of signaling might be connected. For example, could the non-genomic actions of TRs somehow influence those SMAD-independent pathways? Or vice versa.
Wow, that's a really cool thought. It seems like there are endless possibilities for how these different pathways could be interacting and affecting each other.
Absolutely. And that's what makes this field so exciting. Every new discovery reveals more layers of complexity and interconnectedness. It's like we're constantly uncovering new pieces of the puzzle.
It's incredible to think about how intricate and finely tuned these cellular processes are.
It really is. And it makes you appreciate the amazing complexity of life.
Okay, so we've talked about a lot of cool stuff about how cells communicate using these different pathways.
We have. It's been quite a journey through the world of cell signaling.
It has. We explored how AKPs help organize those localized signals in heart muscle cells and how thyroid hormone receptors are like tiny switches controlling genes with the help of their core pressor and coactivator buddies.
Right, and we saw how those examples, even though they seem pretty different, actually share a common thread.
Yeah, they all show us that there are these alternative routes for signaling that can make the cell's response way more complex and fine-tuned.
That's the key takeaway here.
Which brings us back to our main focus. SMAD-independent pathways and what they do in the TGF-beta superfamily. We've been talking about how these pathways, often involving kinases like MAPK and Rho, give TGF-beta more options for controlling cell behavior.
It's like giving cells a whole toolkit instead of just one tool.
Right. So as we wrap up this deep dive, I want to go back to that big question. Why is understanding these non-SMAD pathways so important? Especially when we think about all the different roles TGF-beta plays.
Well, we've seen that TGF-beta is involved in so many different cellular processes. everything from embryonic development and wound healing to immune responses and even cancer.
It's a pretty powerful molecule.
It is, and it can do both good and bad things. So by understanding how these SMAD-independent pathways contribute to both the positive and negative effects of TGF-beta, we can start developing more targeted therapies.
So instead of just trying to turn TGF-beta on or off completely, we could adjust specific pathways to get the outcome we want.
Exactly. That's the exciting part. We're moving towards a more precise way of manipulating these signals, which could have a huge impact on treating diseases.
So basically, we can't just focus on SMA signaling anymore. We need to consider the entire network, all those SMAD-independent pathways too, to really understand how TGF-beta works.
Absolutely. It's like trying to understand a whole orchestra by only listening to the violins. You need to hear all the instruments to appreciate the full symphony.
I love that analogy. It's a great way to think about the future of research in this field.
I agree. We need to embrace the complexity to unlock the full potential of these signaling networks.
Well, I don't know about you, but my brain is feeling pretty full after all that.
Mine too. But it's a good kind of full, right?
Definitely. It's been amazing to explore this intricate world of cell signaling with you.
The pleasure was all mine.
And to everyone listening, thank you for joining us on this deep dive into SMA-independent pathways. We hope you learned something new and maybe even got inspired to explore this fascinating field further. Who knows, maybe you'll be the one to make the next big discovery. Until next time, keep those brains buzzing.